welcome back to the lecture series on cell culture technology. So, this is our third week what we will be initiating, and if you recollect the second week or during the second week we have started the layout design. So, you become a PI or you get a job in the industry and you have to set up a cell culture facility. So, how you will proceed? What are those critical steps which will not be really written in a book, but you kind of have to keep in mind how to move. So, if you recollect back, so one aspect what we have taken into account is the lab is divided into two parts. The part one where some of the kind of dissections which may cause infections or which may cause a lot of microbial contamination should be done in one part, whereas the other part is dedicated for doing the real culture work. So, in that context, if you recollect, we divided the lab into two parts, right. So, another interesting thing which many new modern labs do follow is or rather should be very clearly followed, that they maintain a specific set of footwears. So, say for example, if you remember, I told you that you have a shoe rack and all that stuff. So, you could have very dedicated footwears which could be used for the cell culture facility itself. And apart from it, if you remember where we talked about entering into inside the lab, if you think of this zone where you are entering, there are something called sticky mats. Sticky mats. These sticky mats are basically, if you put your feet, it's kind of, so they are there to, to trap the dust. It's kind of a dust reducer into the system. And most of these rooms are being arranged in a way that the air is being continuously pumped out of these rooms. Okay? So, you have to ensure that the air inside both these rooms are being continuously pumped out and circulated. That means what you are essentially doing is you are continuously recycling the air, and that may demand that the roof what you make, you may need to make the roof a false roof. So, on top of the false roof you may have to put different kind of setup to pump the air. So, these are some of the points what you always have to keep in mind. Even you have to be concerned from the word where you have to talk to your colleagues in engineering who may help you, or in your respective works department of the institute, that how I can have a false roof. Like say for example, I am standing in this room where I know there is a false roof. Okay, so you all have heard about a false roof. It's something like, so this is the height of the room and you just create another false roof using some kind of a polymer, and in between there is a gap, a significant gap there. Okay. So there you put all the pipe holes and everything, suck the air out and circulate the air in a peculiar way. So these are some of the interesting details which you always have to keep in mind. So if we just look through the picture, so you will see there is an entry port here. Then we talked about the sink and the shower. Then we talked about a tabletop autoclave. Here you see the tabletop autoclave. Then you have a sacrificial zone where you sacrifice the animal by using ether, carbon dioxide, cervical dislocation, whatever animal ethics people recommended you. Then you have a dissection assembly where you do all sorts of dissection and everything. Then from here we'll leave the rest of the space. Now you are moving into the second room, which is the room where you will be performing most of the culture work. And in between, I told you that you have a nitrogen storage which has to be replenished time to time. And apart from it, what you can do is out here, I'm using a light green color, from entering from this one to this part, because you have partitioned the room, you can put the sticky mats, so that way that will prevent a further level of checkpoint, or you could have a kind of a sliding door out here which will ensure or restrict entry and the exit along this point. Okay. And there is one more thing which could come very handy. If you can place a refrigerator somewhere out here in the corner, because you will be needing multiple refrigerators, because you have to store mediums, growth factors, several things. So you may think of having a refrigerator in one of the corners and try to use mostly the corners, okay, because that will save a lot of your space. And whenever you design that you want to place a refrigerator, you can have this part, the countertop, maybe cut at that point, and you can place the refrigerator in such a way that you are not consuming that space. Okay, so you'll have one refrigerator out here, depending on what is the strength of the lab. Now from here you move to the mainframe out here. You could have a, if you do not want to put a sliding door, you could have a wind curtain through which you enter here. Now here you have to be really careful, because this is the zone where most of your work will be happening. So I have already told you that this is where you'll be placing the incubators, probably in the corners. So you are not in direct hit of the pathway, and you'll be needing laminar flow hood. You can have maybe 1 or 2 laminar flow hood like this, either. So if this is your countertop, along the side of the countertop out here, but you have to ensure that you are not obstructing the opening of the incubator door, depending on which side of the incubator door, in which side you want to open the incubator door. Okay, so you have laminar flow hood sitting here. So again, if you noticed one interesting aspect, so if I have to like put one single line like this is your entry and cross. So of course there's a wall here, assuming there's a wall or a glass or something, so there's a blockage here. It's not really like moving, but I'm not putting any of the major piece of instrument which will come on the way of the air current. Try to avoid this as much as you can. Do not prefer to keep anything which is coming on the way. It's kind of very widely advised that minimize that, somehow or other minimize it as much as you can. So you have these laminar flow hoods here. You have the countertop, of course, which I am shading in black now. Okay, you have the countertop for you for all other works, whatever. Now you are needed to have whatsoever purpose you have in the lab, but you needed to have a compound microscope, decent compound microscope. Now this is something we will be discussing a little bit more. So, let us get the purpose. So, you have either a cell line what you are maintaining. So, you have to have, I have told you about the nitrogen storage and everything. So, you have to have a storage area for say for example, a storage area out here for storing the cell line, this yellow shading what I am doing. Okay. So, you will have cell line storage, and this is a tank which has to be replenished time to time with the nitrogen as you are every time you are opening it. Now, whether you are doing a cell line culture or you are doing a primary culture, one thing is very critical, you have to observe the cells. As you are passaging the cell or you are plating the cell, you need to observe the cell time and again. So, you have to have a microscope, and I will recommend you, going by the same logic where you want to put it, prefer something which is not coming in the direct way, because you are viewing the cells. So, it should not come on this way. So, your options are on this countertop, somewhere here on this side, or somewhere on this wall, somewhere either this wall or that wall, which one suits your requirements. I cannot tell that, but I can recommend you what all you have to avoid. You have to place a microscope, and this is a microscope which will be probably a compound microscope. Now, whether you wanted to have a fluorescent attachment with it or not. So, you have a compound microscope plus because these are all addendum. That I will leave it to your requirements and to your wisdom, because that decision only you can take whether you want a fluorescent attachment or not. But what I will recommend here is something else, where you are plating the cells. That is important. Why that is important? Because if you are working on substrate which are opaque, so for example, I have a opaque substrate like this, okay, and I plate the cells on top of it. So, it is a tissue engineering, it is a some kind of a hydrogel or a cryogel or some kind of a metal or some kind of a biomaterial, something I am testing the biocompatibility and the cells are on top of it, right. Now, in that case, you will have to have a microscope which is upright. Or, now when we talk about an upright microscope, the thing is that those who have used microscope and those who haven't used the microscope, you have the basic knowledge that in your school you must have read it, that it has an objective. So, you have an eyepiece. So, okay, let us put it like this. So, you will have an eyepiece, you will have a source of light, okay, eyepiece, you have an objective, okay. Now, and you have a source of light. These are the things, and you have a couple of assembly of lens. So, do not worry about that. Now, through the eyepiece, of course, you are viewing the object. Now, your options are two. So, options are this. Say for example, this is where your sample is. Light source may come from top, light source may come from side, light source may come from bottom. Now, if your this is stuff on which the cells are. So, just to visualize it, let me just put it in a slightly different way. Okay. Now, say for example, you have this substrate through which I am just purposefully making it that thick, the light does not pass. So, your only way you can view it is from the top, and you have to shine the light from the top. So, this is how you are shining the light, and the cells are growing on top of it. Okay, and you are viewing the cells, and these cells are these. So, for that you will be needing an upright microscope, means you have this objective which is coming from the top. So, you are viewing from the top, your objective is coming from the top. But the problem with such upright microscopes are the working distance, means this distance between the sample. So, look at this distance between the sample and the objective tip. So, this is the objective lens, and this is where you have the sample, and this distance, let's say I just represent by L, this working distance is critical. Why this is critical? Because most of the microscopes, if you see, will have a very short working distance. What does that mean? That means that L, what you see, what I have put there, L out here, I am just circling it with this distance, what you see, this distance, this L, this microscope, in this case, if it has a very small L, then this microscope objective has to come very close to the sample. Now, think of it, suppose you have a T20 flask, you have this flask on which cells are being cultured, right, which are itself are kind of three-dimensional thing. Now, your cells are sitting out here, you realize, and you have an upright microscope whose objectives are here, and this is the amount, this is the kind of working distance I am talking about, this is the L. So, automatically you won't be able to see much, your visualization will be compromised. But since it is a T20 flask, I will come to the other side very soon, that you do not need the upright in that case. But since your substrate is opaque, it is not allowing any light to come from the bottom, your only option is to use a upright objective in that situation. And for such an upright objective, you have to ensure it is a long distance objective. What does that mean? That means this distance should be higher. So, if this is your sample, you should have the objective this far, and yet you will be able to see it. That is something which no seller of microscope will tell you. They will come and they will tell you n number of info which is to sell their objective. They will not take into account that what are the problems you are going to face. Either you have to be upfront very clear that this is the kind of sample I have to visualize, and you convey them, or you explore it by yourself, because this is a pertinent problem I have seen over the years when we have set up labs. One negligence on our side costed us very dearly, that shit, why we did it like that, we should have done it other way. Okay. So, if you are confident that your samples will not be like this, then you are on a slightly easier side. Then you can go for, so if you know that your plating surface is transparent, light can move back and forth, then you are good to go. Then you can use a objective which is coming from below, okay, from underneath. It is no more upright objective here. So, objective is from the bottom. So, even objective can touch the base of it, because touching the base of the sample which is not in contact won't help. Very simple, say for example, this is your dish, okay, and inside your cells are growing, right, and here is your medium. So, obviously, the microscope can always touch the base of this vessel. It won't, microscope objective can touch the base of the vessel because it's plastic, right, and it's transparent, and you can shine the light through this path and you can visualize it. So, in that case, you have an inverted assembly, your objective is underneath, you have an inverted assembly. Inverted microscope, in that case where the objective is below, you do not have to worry about the working distance. You can really come close to the sample, provided this is transparent. Here this was opaque or translucent. So, your options are very clear. Either, and another interesting part, I used to dream of, but as of now it's kind of not so easy to do. You cannot convert a upright to an inverted microscope. So, if you know that you have two kind of samples which people will be working in the lab, one which will be, say for example, you work some kind of biomaterial coating which is opaque, then you have to have a setup with long working distance. You will have an upright microscope, and you have the other people who will be using on a transparent way. Either you can use the upright one, but the problem, there are issues with the upright one too. Then in that case you have to have another microscope which is inverted. And even with inverted, this is another thing which most of these giant quote unquote manufacturers won't tell. You have to ask them this, and I myself got like kind of, I won't say duped, but definitely was not being told the right set of information, that you have to ask them that, is this objective for plastic or glass? There is something on the lens, objective lens. If you open the objective lens, you will see there is a number which will be given like n is equal to something. I am not getting into that microscopic detail, what does that mean, but that is the one which will tell you that suppose your samples are on a glass dish. So, glass has a different kind of thickness and different kind of optical properties, whereas if it is on a plastic, which is basically polycarbonate, like these kind of T80 or whatever, like vessels, they are different material with a different optical properties. So, they have a different kind of thickness. For that you have specifically different objectives. So, many a times you buy an objective and then you realize that objective is not really the objective you are asking for, because you are unable to visualize. You have a wonderful microscope, but you are unable to visualize things. So, these are some of the points which will never be part of your standard textbook, but this is what comes from experience. So, I will recommend, in terms of the microscope, you be very careful, because this is a big investment. This is not an easy investment. Any good quality microscope costs a fortune. So, I will close in here. In the next class, we will continue it further and we will explain some of the other nitty-gritty details what you will be needing in this journey of learning about cell culture and making it a state-of-the-art facility. Thank you.